myself Dr. Yogesh Joshi. Today we will discuss the basics in pharmacovigilance. So pharmacovigilance is all about the monitoring of adverse drug reactions. Now the contents we covered here is the first definition aspect, second need for pharmacovigilance, purpose or scope of pharmacovigilance, then specific aims of pharmacovigilance, then the heart of the pharmacovigilance, that is adverse drug reaction monitoring, and last we will discuss the reporting of adverse drug reactions. Now first we covered the definition part. Now pharmacovigilance is defined as the science and activities relating to the detection, assessment, understanding and prevention of adverse drug reactions or any other possible drug related problems. According to WHO guidelines, functions of pharmacovigilance are the detection and study of adverse drug reactions, measurement of risk and effectiveness of drug use, dissemination of this particular information, and lastly, education. Next we discuss the need for pharmacovigilance. There are 4 areas where the need for pharmacovigilance has been discussed. First is the unreliability of preclinical safety data. As we have seen, preclinical studies are being conducted in well-controlled conditions. They involve small and specific sample size, and there is a pressure from various groups to reduce time to approval. So these all points will produce the unreliability of the safety data in the preclinical studies. That's why there is a requirement and the need for pharmacovigilance, especially in post-marketing surveillance program. Second aspect is changing pharmaceutical marketing strategies. Nowadays there is an aggressive marketing that is very much deviated from the traditional aspect of the marketing. Second is direct to consumer advertising, and third, many times drug requires the launch in many countries at the same time. So that also creates a need for pharmacovigilance. The third area will be the changing physician and patient preferences. That includes increasing use of newer drugs, increasing use of drugs to improve quality of life, and a shift of supervised to self-administered therapy. And the last area is easy accessibility. We have seen the increasing conversion of prescription drugs to the OTC drug. That is one area that requires the pharmacovigilance. Now easy access by internet. As we all know, everything will be easily connected with the internet. Next is the easy availability of complementary medicine. And last, easy availability of substandard drug. See, these all will create the need for the pharmacovigilance. Next we will discuss the purpose or scope of pharmacovigilance. Pharmacovigilance was mainly concerned with the detection of adverse drug events that were previously either known or poorly understood. So the purpose was to contribute to a scientific understanding of the safety profile of a rather small number of drugs and to advise national regulatory authorities. We have seen during the last decade, the scope of pharmacovigilance has extended beyond the detecting new signals as new kinds of safety concern have now been realized worldwide. That includes herbal areas, traditional and complementary medicine areas, blood products, biologicals, medical devices, and vaccines. So other than allopathic medications, these areas of traditional concern will be of utmost priority in the pharmacovigilance. Now we will discuss the specific aims of pharmacovigilance. As we have also mentioned in the previous slides, the scope of pharmacovigilance continues to broaden and includes a wide range of therapeutic and diagnostic modalities. So, the specific aim of pharmacovigilance as outlined by WHO, that includes: first, improves patient care and safety in relation to the use of medicines and all medical or paramedical interventions. Second is improve public health and safety in relation to the use of medicines. Third, the contribution to the assessment of benefit, harm, effectiveness, and risk of medicine, encouraging their safe, rational, and more effective, including cost-effective use. And the last aim is to promote understanding, education, and clinical training in pharmacovigilance and its effective communication to the public. Next, we will discuss the heart of pharmacovigilance, that is called adverse drug reaction monitoring. ADR monitoring involves following steps. It covers into 4 steps. First is the identification of adverse drug reactions. Second is assessing causality between drug and suspected reaction. Third one is documentation of ADRs in patient medical records. And last one is the reporting of ADRs to the pharmacovigilance centers or other regulatory authorities. Now, one by one, we will discuss the steps. First step, detection or identification of ADR. It involves 2 areas of identification: pre-marketing studies and post-marketing surveillance. First, we will discuss about the pre-marketing studies. Here, the safety and efficacy of new medicine are tested in animal models, and on humans, the benefit as well as risk of new drugs may be obtained by testing. Now, this pre-marketing studies, they have some disadvantages. That's why there is a requirement of post-marketing surveillance. The first disadvantage is they lack sufficient knowledge to extrapolate information collected from the animal studies, directly posing a risk to the humans. Second, fewer number of subjects are exposed to the new drug prior to the general release of product into the market. Third one is clinical trials cannot be done in rare groups of subjects like children, elderly, and pregnant women. And last is the cost reason. Clinical trials often have a short duration, which means they cannot be capable to generate information about long-term adverse effects. Next, we will discuss the second area of identification, that is post-marketing surveillance. And this can be done by different methods like anecdotal reporting, that is case reports. Intensive monitoring studies are also involved in the post-marketing surveillance. Spontaneous reporting system, SRS. Our country will follow this particular reporting system. Cohort studies, case control studies, case cohort studies, record linkage studies, meta-analysis, and the use of population statistics. These all are the various methods for post-marketing surveillance. Next, we move ahead with the second step of ADR monitoring, that is assessing the causality. Causality assessment is the method by which the extent of relationship between a drug and a suspected reaction is established. There are three approaches to assess the causality. First is the opinion of an individual expert. Second is the opinion of a panel of experts. And third is the formal algorithm. These three approaches will have their own benefits. Like first approach, here an expert will evaluate the case. Second approach, a panel of experts will go to a collective opinion about an ADR. And in the third approach, after collecting the data, the critically assessed information using one or more standard algorithm will be utilized. Now, for causality assessment, some of the important algorithms that are being used worldwide, they includes Naranjo scale, WHO scale, European ABO system, Kramer rule, Bayesian approach, Karch-Lasanga method, and French imputation method. But among them, there will be no gold standard for the causality assessment. The categorization of causality assess relationship between a drug and a reaction, it also varies with the scale adopted. Out of any scale above, you can adopt any scale and you can categorize the reaction into different types. WHO scale categorizes the causality relation into various subtypes like certain, probable, possible, unaccessible or unclassifiable, unlikely, conditional or unclassifiable. Similarly, Naranjo scale categorizes the reaction as definite, probable, possible or unlikely. Now we discuss the method of causality assessment. A causality assessment will be analyzed in considering the four different basic points that will be attributing a clinical adverse event to the drug. First, we will analyze the temporal time relationship between suspected reaction and drug. Second, we can go for the de-challenge step, in which the cessation of drug is involved. Third is the re-challenge process, where the reintroduction of the drug will involve. And lastly, we can analyze the likelihood of other possible causes. Now, proceeding with the third step of ADR monitoring is the documentation of ADR in the patient medical records. This particular process will aid as a reference for alerting clinicians and other healthcare professionals to the possibility of causality suspicion of a reaction. And the last step in ADR monitoring is the reporting of the ADR to the pharmacovigilance centers or to the ADR regulating authorities. Different ADR regulating authorities where the ADR reporting can be done, they are CSM, Committee on Safety of Medicine, Adverse Drug Reaction Advisory Committee, ADREC, MedWatch, and Vaccine Adverse Event Reporting System. They are the regulatory bodies and regulatory authorities of all over the world. Internationally, WHO, UMC, Uppsala Monitoring Center, they created our international database that maintain all the data of ADRs. And in India, we have one National Pharmacovigilance Center that is located in New Delhi at CDSCO, Central Drug Standard Control Organization. that is one National Pharmacovigilance Center. We have 2 zonal centers, we have 5 regional centers, and 24 peripheral centers. And the National Pharmacovigilance Center that communicates all the reported ADRs to the WHO UMC International Database. So that is the ADR monitoring. Now we move to the last section of our topic in the basics of pharmacovigilance is reporting of ADRs. Here we cover what to report, when to report, and how to report. That all need to be understood by everyone. So healthcare professionals, they are generally requested to report all the suspected adverse drug reaction to any therapeutic agent, including whether it will be in vaccine or any dental product or medical supplies. And the range from minor reaction to the range of severity called disability or death, they need to be reported. Then second, when to report. So any suspected ADR, they should be reported to the ADR monitoring division as soon as possible. No delay is considered. So any delay in reporting will make the reporting inaccurate and unreliable. How to report? That is also a key aspect of reporting. Here the spontaneous reporting system of adverse drug reaction, we can say, is one of the most effective method of gathering the information about the ADR. So we can go for the spontaneous reporting system. And whenever we've seen any ADR, we need to report through a form that need to be completed by the concerned health professionals and then further sent to the ADR monitoring division. Next we discussed about the ADR reporting form. It consisting of some basic information about the patient, the drug, the adverse reactions, the action taken, and the outcome of the ADR. The following information required in reporting form. First is the patient identity. Second is the description of the adverse reaction. Third, information on the suspected drug that need to be considered for reporting. In the drug information, we need to report the name of the drug, dosage form and its strength, frequency of the drug given, route of drug administration, date of drug administration, de-challenge and re-challenge, whether they will be performed or not, drug which are concurrently used along with the suspicious drug, indication for the drug therapy, treatment given for the ADR happens, and any other additional information we need to be there in the form. This is one of the sample ADR reporting form. It involves all the information about the drug, reactions, assessment, and scoring aspect that is beneficial for ADR reporting. These are some sample formats of some standard ADR reporting forms used by us. Our regulatory body follows these 2 formats for ADR reporting. So these are available online in the official websites of regulatory body as well as the pharmacovigilance regulatory centers. They share the reporting form to all the medical facilities, to all the clinical settings. So here we ended with the basics in pharmacovigilance. Thank you.